Welcome everybody to Biomes. I appreciate your attention. I hope you are all enjoying the sandwiches. We're going to start quickly. Thank you. So again, welcome to Biomes, Bridging Issues and Optimizing Methods in Environmental Sciences. Welcome first years. First Biomes of the semester. My name is Gabriel Gadsden. I'm one of the student chairs, along with Brandon Lewis and some other folks, Anne Francis, Phoebe, Kair. If those names ring a bell and you're interested in getting in touch with Biomes with ideas or joining our committee, reach out to us. Today we have Erick Gagne, who's going to be speaking. I'm going to give a quick introduction. So Erick recently, or not recently, we had a joke about that, actually completed his postdoctoral research in Dr. Sue Van Wood's lab at Colorado State University. His work there uses genomic data to determine how landscape characteristics shape disease spread in wildlife. In addition, the research was investigating disease spillover, transmission of a pathogen from a reservoir host population to a novel host between domestic animals, wildlife, and humans. Erick's work now uses molecular and ecological techniques to better understand population structure of wildlife, as well as the origin and epidemiology of infectious diseases in wildlife. The goal of the work is to further our understanding of the ecology and evolution of wildlife, as well as their pathogens, in order to guide remediation and prevention of wildlife diseases. Please help me welcome Erick Gagne.
Make sure the microphone's on. Share. You're all sweating. All right, thank you very much for the invitation to speak today. I'm really excited to be here. It's my first time at Yale and in New Haven, so happy to be here and get to meet you all and learn hopefully more about what some of you are doing here. And as Gabe said, I'm going to talk. I'm a disease ecologist, and I kind of have a varied background. So when I was thinking about sort of the topic of this seminar series, some of what I do is a little bit merging into the environmental issues, and some is more microbiology. But the focus of this talk, I'm really going to kind of get into how I sort of bridge those gaps. So some of it might be a little bit different than other things, but kind of pull those together. And it is going to be about SARS-CoV-2 to a large extent, but hopefully, as you'll see, I'm going to talk about what we can kind of learn from that and how we can move forward, and not just, I know after a horrible pandemic, we're all tired of hearing about the horrible pandemic, but that continues. So I like to start with talking about how understanding pathogen spillover really requires multiple disciplines. So this is an interdisciplinary problem, really on the science level. Understanding how these occur is a complex process that spans multiple disciplines and fields of study. So this is the sort of famous Swiss cheese plot by Raina Plowright and a group of colleagues that they put out in 2017 that I would like to show, that highlights the different complexities of things that kind of have to perfectly align. Let me see if I can do a pointer here, right? This is showing up on the screen. Okay. Sorry, I know that people online won't be able to see unless I do the proper thing here. But anyway, don't worry about reading the small text on the figure, but really I've highlighted the key areas on here. So you're going to have, right, for a spillover to occur, you're going to have some reservoir host that has a pathogen that exists on the landscape. And that pathogen in that host is going to have a certain epidemiology, it's going to have certain characteristics that really drive where that pathogen is, when there's high levels of that pathogen in that reservoir host, when there's low levels, how it ebbs and flows. And that will, of course, contribute to the probability or the ability for that reservoir host to interact with a naive host and have exposure to the pathogen. Following exposure to the pathogen, you then need to be able to have that pathogen replicate and succeed in this naive host, and then onwardly transmit in that host. So when I think about this, I like to think really that complexity kind of boils down into two real key factors you need to have for spillover to occur. You need to first have an infected host that is in contact with a naive host. And then you need that infection to proceed within that naive host. And although this can occur regularly, it's really difficult to capture this in real time, which makes understanding it or studying it very challenging. And there's some recent work out, again, out of Rena Plowright's lab that I chose to briefly just mention, and you can all read their new paper of Peggy Ebby in Nature that really tells this beautiful story of how changes in the environment interact with the bat reservoir host and cause spillover of Hendra virus in Australia. And the key that I want to highlight here is that this is really capturing almost 20 years of intensive research in a system that has multiple spillover events that has occurred. And it takes, when you have this 20 years of millions and millions of dollars of research, they began to put together and tell this beautiful story. But of course, early on in a pandemic, you're kind of in this more confusing state. So this is a diagram I've taken from someone else that kind of was published early on in the pandemic, where really highlighting the idea that we didn't really know what was going on with SARS-CoV-2, where it came from. We still don't know where it came from. We still don't know what intermediate host it had, or really understand these reverse zoonotic infections where it reinfects and goes back into animal species. So I'm going to kind of talk about a little bit of my perspective and my experience of trying to start to put together some of this mess and learn something. So if you want to hear a beautiful story, someone from Rena Plowright's lab or Rena herself gives wonderful seminars on this really cool system. But here I'm going to kind of talk more about this messy system and how it started. And particularly for this group, I want to talk about a little bit about my background and how that sort of has come together to the way I think about these problems and move forward. So I'll first start briefly by giving an introduction on my background. I like to do that in particular with student-focused seminars. And then I'll talk about how I first started working on SARS-CoV-2 and looking at evolution of the virus in domestic cats and how that's merged into some of the work that I'm currently doing with deer and potential wildlife reservoirs. And then end with talking about future directions and things that I still think we need. There's a lot more that we still need to learn than we've actually learned. So I have a PhD in ecology and evolutionary biology. I went to Tulane University, and I worked in this really, I don't have a time, so I'm pulling my phone out, make sure. Oh, there it is. Interesting system of these endemic Hawaiian stream fishes. So these are stream fish that live in the middle of an oceanic island. And my work was really looking at how this introduced parasite, what abiotic and biotic factors drove loads of this introduced parasite and how it spread across the archipelago. And kind of one of the main takeaway of after we did a whole bunch of genetics of the host, genetics of the nematode, put all this together, and it really came out to that it's a really complex system, and it had to do with different environmental land use changes. Actually, more pristine streams had higher loads of parasites because the intermediate host was highly susceptible to poor water quality. But the takeaway was, even if you can look at a species in every single watershed, we study every single watershed. You have to do these snorkel surveys somewhere in beautiful mountain streams, somewhere in downtown Honolulu, dead chickens and other gross things. So it was really a mixed bag of work. But it took a lot to kind of put together really what I still think was a cursory understanding of what was driving sort of the disease ecology in this system. And I then went and I did a postdoc in a microbiology department, as Gabe mentioned, at Colorado State University, where I worked in a more traditional molecular virology lab, but where they were doing work on natural systems and looking at retrovirus infections in wild felids. And I got the opportunity to join what was a 18-year study. So again, similar to the Hendra story, 18-year study. They had something like $25 million of funding over this time to really try to understand and disentangle the spillover in this wild felid system. And what it came down to, really, at the end of the day, is that, again, it was a lot of different factors. So this is kind of the space I was working in. We were writing the next grant when the SARS outbreak happened, and looking at how, we, or the group as a broad, one thing I didn't mention is a lot of my work and my specific stuff is in kind of genomics of both hosts and pathogens. So that's sort of when you hear these different stories where a lot of where I come in. But how the different, there's different innate and intrinsic immune responses in these cats between different species that really were critical, but also that there was different environmental and land use shifts that really drove those interactions in the first place. So then when the pandemic started in March of 2022, I was working at Colorado State as a postdoc, and I had a meeting with my advisor where we said over Zoom, was 2 weeks to flatten the curve. We said, what are we going to do? You know, we're in a virology lab. We study cross-species transmission, but we really didn't think there was role and we were going to go home. So I went home and I was doing a bunch of coding, enjoying a beer on my deck, watching a beautiful sunset, partially, unfortunately, due to the wildfires. It makes some sunsets prettier, air quality terrible. And then I get a text the same day we actually had the meeting where we're like, we're not going to work on this, to get on a call with a colleague in Hong Kong who had worked at the veterinary school there and was finding a large number, not a large number, but at this enough that it wasn't, you know, double-digit numbers of domestic cats that were infected with SARS-CoV-2. And this is where we had a meeting where Sue in particular, she had a specific pathogen-free colony of domestic cats. Colorado State has a really large animal BSL-3 facility that particularly does weird, like so like a lot of BSL-3, you can do like, you know, they're primate ones, there are rodent ones. This was one where you could do, they've done alligators in there. So you can do different things. And there was a lot of concern on what the next step would be because already, for example, in Hong Kong, they've heard of these reports and there was a lot of sort of worry that people were gonna start throwing their cats out, and that was already happening. And actually, interestingly, we actually found out that the UK, so something I like to kind of put this in context of, you know, other Western governments were considering doing mass calls early in the pandemic of cats. So this is sort of the context where this is working. And then the reason I came in is that Sue said, you know, if we're going to do studies, if we're going to look at and answer these important questions of how permissive are cats to infection, what is the role here? We wanted to sort of leverage and get as much information out of it. So it's important to note, I'm going to talk about the genomic work that then stemmed out of this sort of shift in my career to work on this, that is coming from many other studies, a lot of which was led by Angela Bosco-Loth, who runs that animal BSL-3 facility there. And I also want to highlight that Laura Basher was a PhD student that entered Sue's lab about 4 months after this study started and really has taken a lot of it over as I transitioned out of the lab pretty quickly after, and really was instrumental in helping this. So what we did is we had a series of domestic cats that were infected with SARS-CoV-2. They were then housed with other cats, and we showed that, well, we didn't show, others that I didn't show, others showed that the cats become infected. They get mildly sick, if sick at all. Interestingly, older cats get sicker, so it's like kind of like people. And then they readily transmit to other cats very easily. And then where I came in is I deep sequenced all these viruses. And so when we're talking here, we're talking about within individual variation. So viruses exist as a community within an individual. So you can start to look at evolution and emergence of new variants within that specific individual. So we're going to be talking about variants that arose at a frequency of greater than 3% within one cat. And this is a mess to look at first, but if you look at this blue color here, these are the non-synonymous single nucleotide variants that arose within individual cats. The letters here correspond to different groupings, with here B being the contact cats. And what you can see is that there's a significant number of variants that arose, particularly within the spike protein, which is between these two bars here. And actually, the contact cats had fewer, likely indicating a further additional bottleneck. So we found that there was rapid viral evolution. We found 88 unique variants that was not in the inoculum within the infected cats, many of which were in the spike protein. So we looked at the gene length to the total genome length, and then the proportion of variants within that gene segment. And we found that the spike was an outlier. So there was more variants within spike protein than would be expected based just on the size of the genome. We also found signatures of strong positive selection. And interestingly, we found several variants that sort of matched variants of concern in the human population. We found the D614G, which, if everyone remembers, early in the pandemic, this arose. It's now debated where it arose, but first was sequenced in Italy and then quickly sort of took over and became dominant. And that arose quickly in most of our cats. It was the most consistent sort of variant that arose and actually went to fixation in a cat. So importantly, we're sequencing these 3 and 5 days post-inoculation. So within 5 days of that virus being within that cat, this variant arose. And we found other sort of variants of concern within the cats as well. that match sort of ones of interest in the human population. And we found strong positive selection, but really only in the spike gene. So the other sort of open reading frames didn't show signatures of selection. So then the next question became, what are we seeing in these cats over time? So now we're looking at sort of natural infections where people are now screening, including us. So some of these are sequences, most of these are not, where we're looking at people who bring their cats in and the cats are sick or maybe they had COVID and you sequence them. And these colors here correspond to the major variants within the human population, with red being Omicron, purple being Alpha. And what you can see is that domestic cats, they're infected with every single variant. So we found them in all variants. These are only cats. And now here's a snapshot where we intersperse cats with the most closely related human case. And it's hard to see, but all you really need to see is these big colors here. And what you can see is that almost all these cats were nested within a human cases. So what we're really seeing is that the cats, yes, they become readily infected. Yes, they can transmit to other cats, but that's not really occurring much. Most of the cats that come infected is because their owners are sick or they have a one-off. Which interestingly, this violated all of what I thought was going to happen. I thought feral cat colonies were going to be reservoirs, and we looked for that, and it really didn't appear to occur. So, in summary, domestic cats are easily infected. This is my cat who actually died during the pandemic, not from SARS-CoV-2, but she gets to stay in the talks. But there were no cat-specific lineages. So despite these variants that emerge really rapidly and how you can have selection within an individual cat, because you don't have that onward or consistent transmission between cats, you're not finding cat-specific lineages. So are domestic cats potentially a source of novel variants? Maybe, but because of without that persistent transmission within them, it's sort of less likely. So while we were working on this, suddenly it seemed like every animal under the sun, you just constantly were hearing reports of animals becoming infected. And in the middle of that work, I moved to the University of Pennsylvania, where I now work with the Wildlife Futures Program, and I have a lot of really applied questions. But part of that work, that program is heavily focused on chronic wasting disease, which is a prion disease of deer. So we have a lot of deer samples. So when we started hearing about deer sequences, that took another sort of shift here. But interesting to me in the broader picture again is, so I talked about in the beginning how spillover, you need to have that, that Swiss cheese, that perfect combination of things for a spillover to occur. And suddenly it's, spillover is happening constantly from humans into all of these animals. And I really think that that's because that contact is a really important factor in spillover. The shifts or environmental changes that put new species in contact with each other is, the first step that needs to occur. And once a new pandemic arose in humans, it spread around the globe, and humans have tons of contact with all sorts of animals all the time. So you kind of removed a major barrier. In fact, a lot of these infections, a lot of these species, we only have records of it occurring in zoos, which is kind of one example of that. But you still that allows you to maybe better understand, what's then happening with the virus and the host. And this is where some of the ongoing work is that allows infections to happen, because sometimes their contact occurs, but they're not infected. So at this time, again, I really want to highlight sort of the interdisciplinary and the collaborative nature of a lot of this work takes. And so I had started at Penn Vet, and we, within like a week, I had been getting contacts with lots of folks that were interested in working on the cat problem. And so we kind of had this group going where we were working on the feral cats. And when the deer happened, we kind of expanded out. So this really expands folks from microbiologists in the medical school. Susan YSE is a longtime coronavirologist and lots of experience there. But we also had folks that were, working at ICU physicians and working in the human health spectrum to, the state game biologist who really understands and kind of understands the movement of wildlife. And so we kind of have these different expertise, as you'll hear some about what some of these folks have done. And so when we heard the first sort of early report of deer becoming infected, we began collecting different samples. Now, some of the work and some of our later things work on lymph node samples. Lymph nodes are significant because that's what you collect for chronic wasting disease around, so you have a lot of them. But early on, we actually focused on getting nasal swabs through our we have these 6 wildlife health technicians that did an incredible job of working and integrating with the sort of hunting community as well as roadkill deer and sampling. And we, these 2 students, Haley Zelliff is a veterinary student who ran a lot of the PCRs, and Andrew Marquez helped with a lot of the sequencing analysis. And we found that there was nearly a 20% positive rate in PCR, which is pretty high. In these early reports of deer, the positivity rates in some states was upwards of 40%. And we found both the alpha and delta lineage within Pennsylvania deer populations. And now this is sort of a confusing figure at first, but I'm going to take time to walk through it and show kind of how it shows some interesting findings. So the aspect in having working with the folks at the medical school is that we are actually, at the same time, our group was sequencing thousands and thousands of human cases. So the University of Pennsylvania was leading the sequencing effort for sort of the broader region of Philadelphia and Pennsylvania. And so we have these human cases. So along the sort of x-axis here is the date and time starting. I don't have the year anymore, but this is basically coming into the January of 2022 here from January of 2021. And the color corresponds to the variant. So don't worry about the shadings. These are like, the 1.2.3 or whatever, right? But the basically major variants is the alpha, delta, or omicron, and this is all reflecting the human sequencing. We also pulled human sequencing globally in and from other sources from GISAID here. And then we compared that to the deer sample. So if you look up at the top where our little figures are, this is some of the deer. Actually, haven't updated this. This actually represents like 30 deer, but the general story is the same. But where we found that, we had multiple deer that were infected, and most had the Delta variant, which was what was circulating in the human population at the time. But we found several deer that actually were infected with alpha, but even though that was several upwards of 4 to 5 months after alpha had been replaced in the human population. So when we did sequencing and we did the phylogenies, one thing, so these are different phylogenies. And again, when you look at all these little tiny black circles, these are human sequences that we have from similar areas, as well as 10% from the global population, trying to capture the diversity in the globe at the time. And then the colored circles are the deer sequences. And you can see that you have sort of these 4 clusters of deer sequences in both within the alpha here and the delta. But notably, so that with lots of human sequences in between, which likely indicates that these are 4 separate introduction events, and within some of them you had onward transmission within the deer. And notably within those alpha variants we found that were months after the human, they're on long branches, which represents sort of the evolutionary distance or the amount of change within the sequences from that circulating in the humans. And there's only one human sample that we pulled in that was anywhere close to as diverged as the deer sequences, which likely again indicates that this was circulating within the deer for some period of time without independent of the humans. But we wanted to explicitly test that, so you can use a molecular clock-based approach. So we use a program called BIOMES, which allows you to do time dating, and we estimated that the spillover events occurred in mid-May from a deer that was sequenced. Both these deer were sampled in November, and one was early April and one was likely mid-May. So again, that is when the most recent human sequences were occurring, and it estimates that this is when the spillover event occurred into the deer populations. And then, so we then subsequently, so again, this individual dots aren't important. What is important is colors. And so this orangish color is the deer, where the blue is humans. And this is again, a time dated tree using a similar approach. And what you can see here is, unlike the cats, where you really just had like similar like branches and all the deer all the cats were most clustered with the human sample. Here, deer are clustering together and on these long branches, which is indicative of likely transmission and within the deer populations. And this is from a larger global tree, which includes the Pennsylvania samples as well as others of our collaborators and from GISAID. We also found, I wouldn't say no evidence selection, but limited evidence of selection. But there's a recent paper that shows that maybe somewhat changing. So there is some election in the deer, but not, it's not at the, some of it's in spike, but it's not at the receptor binding domain, which is the like key area that is under selection, for example, in the cats or many other species. So the selection in deer is kind of very different scenario. And just to highlight, that other work has gone on where you've had, significant, this is from a project where we helped collect and contribute to a nationwide sample. So this is going from 30-some sequences to 282, I think. Again, it just shows that human to deer spillover is very common. I think there was over 100 examples of human to deer spillover. There was human to deer. So this is when it spills over into humans and transmits into deer, which is leading to those longer branches. And there was even, this is a typo, human, deer, human. So there was 3 likely indications of a deer transmitting back then to a human. So you can see how, deer are potentially having this virus circulating for long periods of time. The virus is changing, and then it could reinfect the human. Although to date, it's been a rare occurrence. So all this work kind of comes together, but there are, as I think is common in science, there are more questions really than answers. We know it's in the deer, we know it's circulating in the deer, but we don't understand the pathways of transmission. We don't understand if it's being independently maintained in deer. That was all very strongly suggestive that it is, but as you're about to hear, maybe things are changing. And we don't really understand this like limited selection or how the virus is changing in deer, which appears to not be causing phenotypic changes, which means it's not actually changing. It doesn't appear to be changing much of the function or the behavior of the virus. And then an area that I won't talk about today, but it's really interesting to me is, why deer and not elk, and why white-tailed deer at high numbers, but not necessarily mule deer. Like there are these species-specific differences that really aren't explained by the ACE2 or the common receptor alone. And so there are some of these different areas. I think there's going to be a lot to really untangle moving forward, and we're working on doing that. I do want to kind of throw up one slide on some of the sort of more, we would continue with like 20 minutes of microbiology if we were in a different seminar here, but now you're just going to get that this is occurring. Mahan Pereira is a research scientist working with Susan Weiss, and he worked tirelessly actually coming in the field with us some and got good samples and has isolated the virus from deer and has those cultures in the laboratory available, both the alpha, the delta, and now Omicron. And then Elizabeth Lennon is kind of an expert at generating organoid systems or liquid air interface systems for experimental work in VSL3 facilities, and looking at really, can we characterize or look at what these evolutionary changes in the deer mean, and how, that's how we know there's not a lot of phenotypic change from these types of studies that are ongoing. And when we talk back to the beginning of the talk and the complexity of spillover systems, I do think it really takes merging these sort of disparate fields of like wildlife ecology, disease ecology with these like more intensive molecular virology work to kind of get a full picture of what's happening in the system. And then, as I mentioned, we don't really understand what's happening and how transmission is occurring. And understanding transmission in animal populations is inherently very complex. It was a large part of what we tried to do on the puma population, and from that has come sort of additional collaborations on how we might be able to get at this. So when you look at these graphs at the top left, you have GPS movement data. So one way to sort of understand where transmission might be occurring in wildlife species is to track them, see when they have contact with other individuals, and infer that, overlay that with disease dynamics or disease data and change over time. So look at where transmission might be occurring, what the rates of transmission are. A lot of what I do is using sort of a phylogenetic approach to sequencing the pathogen itself and using that information to build transmission trees. And one thing that is really of interest to me and where we're going with the deer project, hopefully, and we've done with the pumas, is how you can overlay these to better understand transmission, because we do know, and we've really found that, missing links are common, right? It's really hard to sample wildlife, and those missing links, unfortunately, do matter. So this is where we're going now with the transmission aspect of the deer, is we're getting together these field studies to look at multiple data sources in a serial sample population. So we want to go out, we want to put radio collars on deer, and then we want to recapture them and sample them over time. And as you can imagine, this is time-consuming, expensive, difficult work. And so actually how this is really coming together is that Kim Pepin, who is a research scientist with the USDA and a disease ecologist, really, but USDA is a big place. More specifically, she's at the National Wildlife Research Center, which is this really great wildlife research group in Fort Collins, but a relatively small group within the USDA. And when the American Rescue Plan put out funding for SARS-CoV-2, they were able to secure a large amount of money to try to bring together multiple individuals to really get at some of these fundamental questions in disease ecology and really understand what was happening within the deer. So I was lucky enough to have a role, but it spans all these different groups. So the USGS Cooperative, for those who don't know, the USGS is actually all the research ecologists and biologists and wildlife biologists are within the USGS due to a restructuring of the federal government a number of years ago. But they run these great cooperative programs. So we're working with the one at Penn State and the one at Montana State University, Colorado State University, Utah State, Columbia, Southern Illinois, University of Minnesota, the University of Tennessee, and then me at Penn. So we're pulling together, by pulling together these resources, we can have multiple populations that you'll see, but we also have a diversity of expertise. So we're grounded in genomics, but there are wildlife ecologists, there are disease modelers of different scopes, there are movement modelers. So there's a whole bunch of different expertise that we're going to try to hopefully pull this group together and really start to disentangle and understand transmission in deer. I did want to sort of throw up some of the individuals' names and just highlight that this is a massive collaborative effort and really kind of this aggressive and really, I think, innovative thinking by Kim to pull this together is allowing this project to proceed. And so what this looks like is we currently have 13 study sites across 7 states where we'll be capturing and doing the serial sampling of deer, because of course, as you all know, being in the School of Environment, understanding and disentangling transmission in like one study site doesn't really tell you much about the whole picture. So we're trying to hopefully get at the broader picture and the broader context by having these multiple sites. And because some of this was new to me, right? Like I have lots of field experience, but I was catching fish in Hawaii or working on pumas where I had limited field work. So I didn't know much about deer capture at the time either. So it really varies by landscapes. You have these kind of complex areas where you bait them in, they're playing, and then this rocket net goes over to catch them. Sometimes you can dart them. There are these things called clover traps where they basically catch them in a cage and then you go in and gently restrain the deer. But to me, that was a wild concept. And they wear hockey masks. Or do helicopter captures, which is really effective in the West, but not effective in the East here, where we have this thick underbrush and really dense vegetation. Even in the winter time, it's really difficult to get into where you could catch them with helicopters. And one thing I want to highlight here, I think, one of the reasons I talked about my background is I had a really different experience, really different environment as a postdoc as I did my PhD. You obviously want to leverage and build on skills and think things together, but I think the postdoc is a nice opportunity to build a new skill set that kind of can allow you to think or do work in different ways. So when I was tasked with capturing a bunch of deer and don't really know what I'm doing, what I did was I hired a postdoc who was a veterinarian also, who had extensive experience capturing ungulates. So Jennifer Oy Pedersen is from Norway, and a fabulous postdoc who has done moose captures, reindeer captures, deer captures, and is really making the field aspect of this work possible. On her side, she's really interested in learning genomics, hasn't had much experience with genetics or genomics, and she's kind of having that lab experience also. But with all new sort of field studies across our board, I'll highlight some things in Pennsylvania. There was a lot of sort of challenges with this work. One was the bureaucracy of the government. We got funded in like mid-February, or like everything was finalized in mid-February to go forward, but capture seasons are like January through March. So we had a truncated capture season where we were adding on to chronic wasting disease work. In a lot of cases, these are also for chronic wasting disease project that I have. That's actually how I originally hired Jen for. And so we had some hurdles getting kind of the samples we needed and figuring out how to bait deer. Also, I don't want to say it's climate change. We had a really warm winter, so they weren't as interested in bait in the East. So like the folks in Tennessee and me had a really hard time, particularly hard time baiting them in. We also had things like I learned, like I didn't know what to do about this, where we had unexpected visitors. So here's our clover trap with the bear. Unfortunately, a bear cub got in. they have remote releases, so it's actually very easy, but we didn't have any deer that day. So from this first year of sampling, as I said, the project started this winter. We have a varied sample size across states as we kind of look at these different challenges. This is it was R1 because it was an existing project for chronic wasting disease and other things, but Kezia did a really wonderful job in Utah getting a lot of mule deer they use. And the other thing is that resampling is very difficult. And you can see we had 4 recaptures in our Pennsylvania site. These sites that have double-digit recaptures were all helicopter sites, so that makes that a lot easier. So we're having to sort of shift some things. But we do have a data set of nearly 500 deer, and we found variable seroprevalence by state and species. Didn't know how much I have some numbers here. So in Pennsylvania, we had 31% were seropositive. I told all the collaborators and ran by the talk, but some of them I'm not going to give specific numbers from their states, but we did find mule deer at 13%, so much lower than whitetail deer consistently. And we had a range of 5 to 40% seropositivity. And excitingly, even with our limited number of recapture, we had deer that were seronegative that converted over the study point. So we saw deer that were seronegative when we captured them the first time. We recaptured them several months later, and they had become positive. I should also note that a lot of our recaptures are kind of just starting or ongoing now. But I'm the geneticist. My role was going to be doing the genetic work. We had found, 20% PCR positivity. There was hope for this, but nationally, we just found a huge reduction. And this is across the USDA's study where they worked with state agencies to get and look at lymph node samples. We've contributed a lot of these too, and just found that the PCR positivity in deer has plummeted to below 5%. And we got goose egg. So in all of our 500 deer, we had no PCR positive deer. So that's another area of shifting. But it also is another area of like extreme interest to me. So one of the things anecdotally, which I meant to point out in that phylogeny, but like we're not seeing Omicron in high levels. So it's, have people heard that deer become infected and change behaviors? Is there a high level of immunity in deer? Deer only, reproduce once a year. There's only naive individuals being introduced in May. Or is there something in the change in the virus or some combination of that factor? So we don't really know why these PCR are plummeting. But those few seroconverted individuals are also really interesting because the PCR prevalence in deer was exceedingly high. It shouldn't have been that high, right? It was like weird to us. And with a few of the lab studies, what we were seeing is that deer just stayed PCR positive for a really long time, even though they didn't stay infectious. So they said you can do something called a plaque assay or different assays to see if you're able to recover infectious virus. And deer, similar to humans, only stayed infectious like 5 to 10 days. But they would have PCR positive results 4 or 5 months later still. And because we had those individual seroconvert that were PCR negative both times, there may even just be a shift of how much time, how long the RNA is persisting. So moving forward from there, we plan to greatly increase our sample size in PA. We've added another site, this Treasure Lake community, where you can see the deer are habituated, which should make them easier to capture. We'll gently dart them, sample them, release them. They'll go back to eating in someone's front yard happily. We've added a new state, but we're also, where my shift in my focus is really going, is looking at potentially using other pathogens or other markers to untangle and provide that piece of the transmission story, to look at just in generally how do directly transmitted pathogens transmit amongst deer populations. So to kind of bring it back to the beginning, forecasting spillover, as I said, is very complicated. And I think, from this is like a diverse area of work that I kind of tried to do some highlights of how we sort of grapple with coming together of trying to understand this as it occurred sort of in real time in these animal species of this human pandemic, really in our backyards here in the United States. And, to really understand it, it's going to take pulling together these different aspects. What is happening and changing with the pathogen? What is happening that drives transmission? Why is it, going from 30% PCR positive to 1% PCR positive? And I think, these types of studies are hopefully what we can come from this is that we can start understanding and disentangling these processes, which will allow us to just understand disease dynamics and transmission of wildlife better, and maybe have us better prepared or better able to think about sort of the next pandemic. And as this one continues, right, there's still the possibility, like, I don't like to be alarmist. I think my side, I think more about sort of the academic questions that we can answer from these systems, that it's just we're watching a new species, a new virus infect new species in real time, and we can learn a lot from that. But there is still the possibility that, deer will become a reservoir of this, horrible virus in our own backyard. So monitoring and understanding that and developing understanding of what's happening to deer is also, could have human health implications. Oh yeah, there's a bit, because now we're kind of focused on that bottom section of that chart. And I think something I want to highlight here is really that a lot of this work was really interdisciplinary, right, where we combine these different disciplines that work together, and it's so critical. And it was interesting as it starts to, have the, wildlife ecologists at the table with the microbiologists, right, where they're coming up with these ideas about how this, what they're seeing in the lab could be occurring. And it just doesn't make any sort of biological sense with the reality of like the ecology of deer or what's happening with deer, The one, I won't say names, but one of the microbiologists asked if there was, even 100,000 deer in Pennsylvania when there's like 3 million probably, but we don't actually know. But something I love about this group, and when I was reading more about what you all do, is that, this is just with like a small subset of understanding the science of what's happening in these animal species. But this new term, this transdisciplinary, where you really kind of create new disciplines, right? There's so many factors outside of the science that need to be integrated and thought about when we think about this. Like waiting, I've never worked on something that sort of had political connotations, and unfortunately this pandemic has become politicized. So understanding and navigating sociology, political law, what should we really be doing, how do we actually implement and translate science as it occurs in real time? I think there's a lot of exciting aspects and things that need to be done, but from across a slew of environmental problems. And so with that leads perfectly into my acknowledgements, where I just have a large slew of names. This took a lot of work, right? We are catching deer in 7 states. We're doing BSL-3 laboratory experiments. So lots of people contributed to all this work. And it's been exciting to be a part of it and learn from all these different people and different disciplines. And with that, I am happy to take any questions. Yes. Mm-hmm.
That happened in the spring. Is there something about the spring regarding things? Second question. Sorry to pass that. Is that from having or problem
in the deer population? Yeah. So the first question of what was occurring in the spring, there was transmission at other times. That was that transmission that was so notable because it was so long before we sampled it. But that transmission study is really trying to get at that big question of where is this transmission occurring. And I actually forgot to mention, and I like to point out on that last deer slide, a lot of people thought it was maybe the hunting community or the different outdoor enthusiasts, but people have a lot of interaction with deer. And this picture is like one on I saw this occurring, and then they had it on their website. So this is a orchard that is outside of Philadelphia that has hundreds of thousands of visitors in the fall. And this is a child sharing her hoagie with a deer. And when I was there were wild deer sleeping right next to the pen. So I don't know, I don't really, springtime's a nice time. People have more contact, but people in rural areas, they feed deer a lot in the winter. Like there's lots of interaction with deer. And I think really what I'm saying is there's just a lot more. Like, I don't think it's going to be one theory. At one point there was like, is it wastewater, which could be a thing? Is it, whatever. So yeah, I don't necessarily think there was something in the springtime other than maybe it was nice weather to be outside. Do deer get sick? So there is no evidence that it causes much pathology at all in deer. So in the experimental infections, they do quite well. Most experimental infections have been with fawns, and been relatively limited because I think at this, having deer and BSL-3 is difficult, and also for like an animal welfare thing, but there aren't like, and maybe as many questions now we know they're infected, but it does appear to have limited pathology in experimental settings. In the wildlife, like one of the things that's so hard with wildlife is, you know, if they're not dropping dead in droves, it's really difficult to figure out what influence it has on them. So, like I know one group that had collared deer out during the peak in deer infections, and they said that their deer moved like some massive percentage less, but that doesn't seem to be replicated in other studies. But so there's some kind of anecdotal evidence that maybe there is sort of some pathology in deer, but it's really not understood well. Yes.
Hi, thank you so much. That was great. I have a question about, you mentioned at the very end of your talk, you're looking at sort of a larger span of diseases across deer populations. And I'm wondering if there's any evidence so far, if you have any thoughts about co-infection or comorbidity, like, is there some suite of characteristics that might make deer more vulnerable?
Yeah. We don't have any evidence of looking at co-infection or making them more vulnerable. It is something we sort of teased apart a little, started to look at a little bit, and so far that it doesn't seem that there are, but we haven't looked at a large scale, and that's something that we're sort of starting to work on now. We're interested in that with like other aspects of deer disease too. There's a large deer die of pneumonia a lot, and they don't really know why. So that's also being channeled into some of this. Yes, Andrea.
Do I have to? Yeah. Okay. So one question I had was the possibility of an intermediate host considered for deer?
It was considered, and there are groups now that will probably be getting at that. So there's a group at Penn State and a group at Virginia Tech that have large pots of funding to essentially look at all the animals and what's out there.
Mm-hmm.
The sequencing analysis, like we think that it's not an intermediate host, but yeah.
And then my second question was whether or not co-infection, she kind of brought it up, with CWD might predispose deer in a behavioral sense to come more in contact with humans.
Yeah. So that is the one thing we did look at. So we looked at co-infection of CWD because we had a lot of, and with the data set that we currently had, which was relatively a couple hundred individuals, we didn't find any relationship between chronic wasting disease infection and SARS-CoV-2 infection.
Okay, thank you.
Yes. So I'm not fully up to speed on it, but from my last time looking at it, there has not been spillover to cattle, and it's likely, I'm trying to remember the study in details, but basically I think cattle can't become infected, or at least if they are infected, it's a one-off. I don't remember if they managed to infect them or not at all, but so it seems like, you know, that's kind of that interesting thing, is where we have that contact. Clearly cattle are in contact with humans all the time, but the virus seems to be unable to infect them. Yes.
Okay, I'm gonna jump in with a question.
Yes.
Evidence of trophic transmission. Do you know anything about either consumption patterns from humans consuming infected deer, or infected deer being consumed by other wildlife in the system? That would be a form of transmission.
Yeah, so in terms of trophic transmission to humans, you know, we don't have any. I guess one of the human infections from deer, they did have close contact, but we don't think it's from consuming the deer, but rather having that close contact with the carcass. Because other studies have shown, you know, cooking your food would kill the virus, or freezing it also would. They have found mountain lions that have SARS-CoV-2, and I think it's from eating deer, but we don't have actual scientific data to back that up. It just seems like mountain lions eat deer, and that would make sense. So I do think that is occurring, but that's just a hypothesis. And also for folks online, I can't see, maybe if it's not, I can't see the chat or anything like that. So if you have questions, just jump in. Yes.
We learn a lot about how changing climates is resulting in species migration into different areas, especially urban areas. So I'm just wondering conceptually how you see your field of work changing or developing in the coming years as we face these like more in human wildlife interactions.
Yeah, so I think like not necessarily what I'm personally doing, but the field in general is really going towards, and there are people here that are working on that, towards looking at how these interactions, particularly in urban, but also ex-urban environments, which is more like, sparser populations that are really common in the West now, and how these are sort of shifting disease risks and transmission risks. Interestingly, which I didn't include this, did a heavy cut of this, which I actually made it right on 45 minutes talk before, but there was a slide. So we don't find association with the deer infections and sort of urban environments, which is interesting, but certainly the more contact that humans have with wildlife species, the more of these sort of potentials that occur. And I think that's going to be a major area of research and study going forward.
All right, can we give Dr. Gagne another round of applause?
Thank you.
Again, Dr. Gagne is going to be on the third floor hanging out. So if you have any questions or just want to chat, do that. For the PhD students in the room, 5 o'clock, Marsh Hall is going to be around. I also didn't thank our wonderful admin staff, Melanie, Lori, and Joan, for all the work that they do. So can we give them a round of applause too? And again, if there's any questions about biomes or any ideas, feel free to let us know every Wednesday, 12 o'clock. We'll see you next time. Thank you. Great talking.